You want healthy, glowing skin, but with all the products, trends, and conflicting advice, it's hard to know what actually works. I understand. I am Dr. Tiina Meder, a former cardiologist and specialist in the skincare development and cosmetic safety expert. I've spent years studying the science of skin. Welcome to Meder Beauty podcast, where we uncover what really matters, break down the science, and give you real expert-backed skincare advice. Whether you're looking to achieve radiant, healthy skin, curious about anti-aging, or want to understand what's really in your product, you are in the right place. Rooted in science and the truth about what your skin actually needs, we'll have in-depth conversations with leading dermatologists, medical experts, and skincare brand founders to bring you the latest innovations, evidence-based insights, and industry secrets that truly make a difference. We are so happy you are here. So let's go! Me personally, I think that the skin is the most fascinating body organ. I know how it sounds. I work with the skin, so I love skin more than anything else. But actually, the skin is truly amazing and surprising. Yes, it is the largest body organ, everyone knows about it, but it is also one of the most diverse human organs because in the skin, we have many types of cells. And this diversity, it's almost unprecedented. Why it's so important to understand skin cells type and why it is important to know about different types of skin cells? Because different skin conditions, they are involving different types of the skin. So when you're trying to treat something or correct something, it can be pigment spots, it can be wrinkles, it can be skin dryness or some elasticity concerns. You should understand what type of skin cells you address, because it defines your choice. Skincare products of different ingredients and also different modality, because laser interacts with one type of cells, microcurrent therapy interacts with another types of cells, and so on. So you should really understand what types of cells you have in focus now. How many skin cells type we have? The simple answer is many. To understand how they function, first of all, I remind you about skin layers. Let's go simple. We have three big skin layers. First, the superficial layer called epidermis. It is our protective outmost layer of the skin. In the middle, we have the layer called dermis. It is the structural layer responsible for the elasticity and for the skin tonus. And finally, the deepest skin layer, which is very often forgotten by beauty specialists, it is the hypodermis, something which we down under the dermis and the epidermis. Hypodermis is basically from the fat tissue. So hypodermis is probably the easiest skin layer because in the hypodermis, the main types of the skin cells, they are fat cells or adipocytes. We have some nervous cells there and we have some immune cells there, but the key cells type, they are fat cells. In the dermis, we have key players called fibroblasts. And when I've been speaking in the podcast about the skin elasticity and collagen, I've been speaking about fibroblasts, and they're very interesting. So they are very interconnected players, extrovertive and optimistic skin cells. But also in the dermis layers, we have not only fibroblasts, pigment cells or melanocytes, they are also located in the dermis. And we have one player which play for the immune system, defense team, they are macrophages. And another immune system players, which is responsible for the inflammation and allergic reaction. They called mast cells or mastocytes. If you are going even higher in the superficial layer of the skin, you are in the dermis. And in the dermis, we have two main types of the skin cells. They are keratinocytes, or sometimes they are called now keratoblasts. And another type, they are keratinocytes, or keratocytes. As you can probably guess by the name, They are related to each other. That's the same family. But in the epidermis, we have also two other types of cells. First type called Langerhans cells, and they are playing for immune system. And another type of cells, they are small mechanic nervous sensors called Merkel cells. So let's count how many cells types we have in the skin. We have two types in the epidermis, two key cells, and two additional cells. We have four types in the dermis. One is the main player, fibroblasts, but also pigment cells, and two types of immune cells. And we have fat cells in the deepest layer of the skin. So 9 skin cells types, and all of them are different. Let's start from the surface. Epidermis is the very varying skin layer. In the epidermis, we have 5 separate layers, and first of them called basal membranes. And this basal membrane is populated with keratinoblast. There are no keratinocytes there. Keratinoblast, they are kind of mother and fathers of keratinocytes. They are parents of these skin cell types, and they are constantly dividing and constantly proliferating. What I find very interesting, they are relatively long-living cells, and we have all the time, we have new keratinoblast in the well-functioning basal membranes area. In these basal membranes, we have these keratinoblites which are dividing all the time, and this divided keratinoblast, one of them is always migrate in the more superficial skin layers. And once it's divided and started this migration, we call it keratinocyte. And what is the biggest difference between blast and cyte cells? Blast cells is constantly proliferating. Cyte cells is not proliferating at all. It doesn't have children. It's child-free cells. But it is the cells who love to travel. It's the traveler cells. And it's going in this big travel from the deep layer of the epidermis to the surface. This travel has kind of constant timing. Probably you all heard about this 28 days cell cycle. And this 28 days, it's most of the time, it is used as an average cell's life, but it is not the life of all types of cells in the skin. Sometimes I met beauticians who think that every skin cells lives 28 days. First of all, it is very average. So we don't know really how long each can live. It's an average timing. It can be from 20 till 40 days in normal, healthy, relatively young, functioning skin. And in these keratinocytes travel to the surface, transforming itself in this travel. It's like, the human being. When you travel, you meet different people, you have a lot of different emotions, and you change traveling. Something like this happens with the keratinocytes. In this travel, first takes all this biochemical experience inside. It synthesizes different substances, but somewhere in the mid of the epidermis, it starts to release and liberate different substances. For example, it is full of ceramides, special lipids, and when it's going to the surface, at some point, the keratinocytes just open the door and release the ceramides in the intercellular space. Finally, we find the ceramides as one of the most essential elements to build and maintain our skin defense system, our lipid barrier. Ceramides will be mixed with lipids produced by the sebum glands, by sebocytes, cells of the sebum glands, and will create this multifaceted, multifunctional lipid mantle of the skin. Also, in the keratinocytes, we can find different kinds of very important proteins. We can find different types of amino acids. For example, one of the proteins, filaggrin, it's based on the amino acid called histidine. And filaggrin, it is the molecule which works as a water-holding magnet in the meat of the epidermis. So water can be held not only on the surface of the skin, but also in the deeper layer. And one of the mechanisms, it is this protein produced by keratinocytes. Another very interesting protein called elastin is liberated a little bit more superficially in the high layers of epidermis, and elastin is one of the most beautiful proteins you can imagine. It's opalescent, really pale rose, and it creates this aura of the glowing skin. Keratinocytes in the very young skin produce a lot of this elastin, and it reflects the light much more efficiently than the rest of the structure of the skin. And as you probably remember from different episodes of my podcast, then skin optic and skin appearance really defined by the ratio of light reflection and light absorption. Elastin reflects the light and creates this youthful appearance of the skin. So I can go on and on keratinocytes' work in the skin, but you should understand that they are very important cells, but they are most fragile cells of our skin. Why? Because they're located very superficially. So every time when you use some aggressive treatments or some aggressive movement against the skin, when you're using aggressive ingredients, they can damage these keratinocytes. And also you should understand that this timing of keratinocytes lifespan and keratinocytes travel is very important. In the young and healthy skin, this timing is between 20 and 40 days. But when we become older, our skin cells travel longer, but in the same time, they travel less successfully. Everything exactly like in the human society, right? And when our skin cells travel on the skin surface, when you are in your 60s or in your 70s, it can take 60 days or 70 days. And when they're coming on the skin surface, they look different. They have less of essential substances, they have different shape, and they stay longer on the skin surface, so they can be mechanically scratched or mechanically simply dusted or damaged by the air pollution, water pollution, or the ultraviolet more than younger cells, which leave the skin surface much faster. And also, it defines the skin appearance. It plays with the skin texture. But as you remember, in the superficial layer of the skin, in the epidermis, we have not only keratinoblast and keratinocytes, progenitors and children, but also we have two different types of skin cells. First type of skin cells, they are Langerhans cells. They are cells of immune system, which create so-called innate immunity of the skin. The role of Langerhans cells is to know as much as they can of different microorganisms, especially bacteria and fungi. And Langerhans cells, they're interacting a lot with the microbiome. Since the very beginning, since the very minute of our life, our skin microbiome acts as a very attentive teacher, which interacts with Langerhans cells. And it teaches Langerhans cells to recognize different kinds of pathogens, to recognize and differentiate micro-friends from the micro-enemy. In the healthy skin, Langerhans cells can recognize easily pathogenic microorganisms. And okay, we have some infection of the skin, Langerhans cells signals about this infection, and our immune system starts to act, locate infection, build this anti-inflammatory wall around this place when you have this trauma and infection site, and start to repair this inflammation. And our immune system starts to act, trying to repair all this inflammation, trauma, and infection in our skin. If Langerhans cells are not taught properly, for example, the skin microbiome was somehow damaged, It can happen when there's been some disinfectant used on the skin or some aggressive treatment used on the skin. And it is especially dangerous in the very early age. When baby is born, it needs to have contact with the external world. It shouldn't be cocooned in the sterile environment because it can damage the microbiome and it can really impact the whole life, creating these holes in the immune system recognition of pathogens and impacting the skin innate defense system. But also it can happen in adult ages as well, because our immune cells, they're constantly learning from our microbiome. The role of the microbiome, amongst other, it is an adaptation of our skin, of our body, to the new environment. So it is very important to keep our microbiome happy, balanced, and do not aggress it, do not disinfect it, do not try to sterilize the skin. So disinfectant are the big no for now, except for the surgery block, probably. In all the rest situation, you should be in the contact in the external world, and it helps your immune system to recognize pathogens, avoid infections, but also moderate your immune response and allergic reaction to everything else. And another type of skin cells which we have in the epidermis, they are Merkel cells. They are coming from the nervous system. So you see we have three different sources of cells in the superficial layer. We have skin itself, keratinoblast and keratinocytes. We have immune system cells and we have nervous system cells. Merkel cells, they act as mechanic receptors in the skin, mostly. They are located in the basal membrane, in the basal layer, next to the keratinoblast, and they are reacting to the pressure, to different kind of mechanical aggression, and signals around them to help skin act properly. When you have some pressure on the skin, you have the whole spectrum of reaction in our body. You feel something, it can be the pain, it can be just the pressure. You can see something, your skin, it can redden. It depends on the reaction of your vascular system, of your capillary. So the whole spectrum in reaction happens in the skin, and most of them, they happens because the small Merkel cells send signals to different structures, to your capillary system, to your pain receptors on the skin, to the brain, but also in the deeper layer of the skin. Because if the skin will be traumatized by the pressure, you need more of collagen. You need your keratinoblast cells divide more efficiently. So all types of pressure can be therapeutic or can be damaging for the skin. And by the way, when you think about massage, using just manual massage, but also different types like gua sha massage or massage with different devices. And why it is so interesting about Merkel cells? Because Merkel cells, they are very much involved in the reaction to such a classic aesthetic procedures as massage. Sometimes I see the people, they are, if not skeptical about massage, but they are speaking about massage like it is something pleasant, psychologically nice, relaxing, but it doesn't have any physiological impact on the skin. Actually, it is very much impactful. And when you proceed massage using your fingers, your hands, or different kind of devices, for example, gua sha, why not? You act as a mechanic pressure on the skin, and these mechanic pressures involve Merkel cells. Merkel cells send signals to different skin structures, and it can really activate the fibroblasts. And we have some scientific research showing that regular massage of the face and body can improve skin elasticity because it activates fibroblast work, and it helps to produce more of collagen, more of elastin, and more of hyaluronic acid. It helps to restore and maintain the skin defense because Merkel cells, they're interacting with keratinocytes and keratinoblast as well. It can impact the activity of pigment cells as well. And here probably is most important to then pigment cells can react to mechanical pressure. So all type of regular strong mechanical pressure can create a hyperpigmentation of the skin. And obviously, massage and pressure interact with the capillary system, so you can improve the microcirculation. It helps to oxygenate the skin, nourish the skin, bring more of nutritional substrates to the deep skin layers and also to the superficial skin layers. But also it can liberate the skin. It can eliminate different toxins. It can eliminate the excess of the water. So massage is very efficient, and Merkel cells, they are most important sensors which works when you mass the face of the body. If you go deeper in the dermis, you will find this almost solid structure built from three-dimensional helix of collagen fibers, steel-like structures of elastin fibers, and all around the cells and around this endless fibrous structure, you will have this matrix-like gel created with a hyaluronic acid. And all the structure is created by one type of cells called fibroblasts. Fibroblasts, as you can imagine, they are a bit similar to keratinoblast because they are progenitors of all the structure. But fibroblasts are not dividing so actively as keratinoblast. The biggest difference in keratinoblast can divide all our life and multiply all our life. Yes, they act slower in the older age, but they're still acting, they're still dividing. Fibroblasts, they are very actively dividing in the young age and early age. And after they slow this process and they almost stop to divide at the age of 25 years old, it doesn't mean then the aging starts at the 25. It is a young age and it's no destruction around the cells. But fibroblasts produce less of elastin at this point, but also they destroy less of this element. Because the key things to understand about the fibroblast, they are cells with double-sided functions. They are builder cells and they are destroyer cells. At some point of the life, they build and destroy with almost the same intensity. It happens in the children's skin and the very young teenager skin. So we have all the time these fresh structural elements in the skin. But in the older age, they build less but start to destroy more. And that's why we lose the skin elasticity. We start to lose collagen because fibroblasts become more destroying than building. They just change the personality and change the character. Once again, so anthropomorphic, exactly like human being. Yeah, it's in the old age, they can become less nicer. And fibroblasts, they are very interesting cells. They have this long life. They are living decades. But anyway, we start to lose them, and we start to lose them relatively early. And it depends not only of our genetics, but also depends on external factors. That's what we call the extrinsic agent, because when we start to lose fibroblasts, obviously it triggers and activates aging processes in the skin. In the long lifetime, from our 20 till our 80 years, we can lose until 35, maybe 40% of fibroblasts. But the rest of fibroblasts, they stay in the skin, and despite changing, they stay more or less functional. In the young age, fibroblasts, they look like big stars and even maybe like starfish. Because the mechanical tension, it's very important for the fibroblast functioning. So fibroblasts stick to collagen fibers and elastin fibers around them. And in this position, it's constantly pulled off and pushed inside. And only in this position it can produce enough of necessary structural elements and can function properly. Once collagen is deformed somehow, for example, by the ultraviolet or by stress hormones or by the air pollution toxins, fibroblast collapses. And when fibroblast is collapsed, it cannot produce the structural elements anymore, but it still can produce destroying enzymes, so it's become more destroyer, and that's how aging process is in the skin. So everything which can deform collagen, which can damage collagen fibers in the skin, will damage skin cells. Why it is so important to understand? Because I see very often in the aesthetic medicine, we have a lot of new methods right now. And many of these methods, they are focusing on the fibroblast activation. So we can use different kind of energy-assisted devices which heat the skin, and this heating activates the fibroblast. We use sometimes injectables which address fibroblast also as an activation process and so on. But these activators of fibroblasts cannot activate them if they are collapsed. When you activate the collapsed fibroblast, it destroys the skin. We want him to build collagen, but paradoxically, this external activation just worsens the situation. And in the long-term consequences, we see even more of skin elasticity lost. So we should be very careful in understanding better what is going on the skin before using any kind of activating methods or treatments. Also, in the dermis, just neighboring fibroblasts, we have very interesting skin cells called melanocytes. Melanocytes, they are pigment cells, and they are living even longer than fibroblasts. They also have this amazing lifespan for many decades of life. And the melanocytes, they are long-living cells, but instead of producing structural elements, they produce mostly pigments. Also, we are losing melanocytes during the lifetime, and we are losing about the same percentage of melanocytes as fibroblasts during our life. But what is the main difference between fibroblasts and melanocytes? They are not related to each other. Fibroblasts, they are cells of skin itself, and they are very much involved in all kinds of conjunctive tissue, because everywhere when you need collagen or elastin or any structural elements, you will have fibroblasts. But melanocytes, they are born in the structure called nervous crest during the embryonic development. And in this embryo development, melanocytes migrate into the skin, but they are not skin cells. They are cells of nervous system, just working in distance from the skin. They look like neurons. They have exactly the same biochemistry as neurons. Only difference with neurons, they are able to produce pigment called melanin. And we have melanocytes in the skin, we have melanocytes in the hair follicles, we have melanocytes in our eyes, they define the eyes color, and we have melanocytes in the nervous system as well, including our brain. And all this interplay between nervous system and skin cells is very important. So if you understand then new pigment cells are nervous cells and not skin cells, you will understand better why when you are stressed, you have a higher risk of the hyperpigmentation. You understand better why the ultraviolet, why sun rays not only affect the skin, but also affect your mood, affect your nervous system as well. So melanocytes, they are such interesting travelers who react not only on the external factors, such as ultraviolet. Obviously, it's the biggest trigger for melanin production, but also they react to the inflammation. They react to the stress hormones. They react to sex hormones, such as estrogens and androgens. They react to mechanical pressures. Every time, for example, when you wear the same spectacles every day or the same sunglasses, and when they are a little bit heavy, it increases the risk of hyperpigmentation on your nose when you have mechanical pressure from your glasses frame. Because melanocytes, they will react to this. They interact with Merkel cells as well. So the main triggers of pigmentation or hyperpigmentation. And these triggers should be addressed not only by ingredients which are able to interact with skin itself. You should understand that every time when we address melanocytes, we address nervous system, we address the brain. And sometimes we should start from the nervous system in the pigmentation treatment. Also, we have another neighbors of pigment cells and fibroblasts in the dermis. One neighbor, they are mastocytes. They are cells responsible for allergic reaction because they're able to release histamine in the skin. And that's how you react to the insect bites. That's how you react to some contact allergens. So when you're using some kind of new skincare types, if you have a reaction, if you have the contact dermatitis, redness, itchiness, some kind of irritation of the skin, or even some kind of elements in the skin, it is the reaction of the skin layer called dermis and of the mastocytes in the dermis. And histamine produces this inflammation reaction all around them. And in this inflammation reaction, we can trigger accelerated aging process as well. So if mastocytes are constantly activated by some toxins, by some allergens, or by some aesthetic aggression in the skin, you can get the chronic inflammation of the skin, which is very dangerous for the skin health, but also it's the main trigger of the skin aging process. Also, inflammation affects melanocytes function in the same time. Every time when the skin is inflamed, every time when the skin is sensitized, it increases the risk of hyperpigmentation because impacted melanocytes start to produce more and more of melanin granules. And hello, hyperpigmentation in the area of contact allergy or in the whole inflamed area. And another types of skin cells in the dermis, they are macrophage. It is a second line of defense for immune system. You remember we have microbiome on the skin surface and living in different niche of the skin. Now we understand that our microbiome, bacteria, fungi, and especially viruses, they can live not only on the stratum corneum, the most superficial layer of the skin. They can live deeper in the epidermis and they can interact with the dermis as well. But in the dermis, if we have some penetration of pathogen in the dermis, there are macrophages who are acting, it's called phagocytos. So it's literally eat the pathogens. It just opens the big cell's mouth and take all pathogen inside of the cells when it digests the pathogens, destroying them. So they're very important for the skin functioning as well. And finally, if you go even deeper, you will find fat cells, which creates a cushion for the skin. But also fat cells interact with other skin cells type, and especially with fibroblasts. They have this very complicated relationships. So if skin cells will have some social network, fibroblasts and fat cells will be like, okay, we are in complicated relations. Because fat cells can command fibroblasts to be more active or less active. How it happens? When our fat cells change the size, so you take weight, you have more fat under the skin. They send signals to fibroblasts into the skin dermis, and literally they signal them, produce less of collagen, produce less of elastin, because we need more space. We need skin to stretch. We need to really increase this surface. And that's what fibroblasts do. They change the collagen production and they start to produce more elastic, soft collagen. So fat cells can take more space and feel more comfortable. So when you take weight, your skin becomes less elastic. Your fibroblasts become less active. And also overweight and obesity, they are pro-inflammatory condition, inflammatory skin cells, and immune system will be reactivated as well. But when you lose weight, when your fat cells become a little bit smaller, they send other signals to the dermis, they send other signals to fibroblast cells, and they ask them to produce more of collagen and more of elastin, which creates this absolutely natural skin lifting reaction. But the thing is, then you should not lose the weight too fast, because fibroblasts will be reactivated gradually. So when you're using some kind of modern weight management, sometimes it acts really dramatic, and that's why we have this phenomenon called Ozempic phase, when you lose the elasticity of the skin, and it can lead even to the face deformation. So you should be very careful. And when you lose weight, when you manage your weight, you should lose it gradually and start slow and comfortably for the skin. But the good news is then, yes, in the older age, our fibroblasts will be less active. They are less able to produce the same amount of collagen, but they still can do it. So you can manage your skin elasticity interplaying with your fat tissue. Why fat tissue is so important? Because it's also the source of stem cells for the skin. It is the source of the new fibroblasts. Another source of the new skin cells, functioning skin cells for the epidermis and for the dermis, it is hair follicles. So it is important to keep them active, as well as the sebum glands. And when you try mattify the skin, when you try to reduce the sebum gland functioning, for example, in the oily skin, it can be a bit risky and it can be a bit tricky, because it can reduce the collagen synthesis as well. It can reduce the fibroblast activity. It can play different things with the epidermis. So keratinoblast can renew slower or faster, and it can really impact the skin appearance and the skin health. As long as you have fat tissue under the skin and your face and your neck, you are good, because you have this stock of stem cells, so your skin will be able to take them when needed. Why this information is also important? Because sometimes we use different types of aesthetic treatment, damaging the skin and trying to make our skin cells to repair skin and to produce more of collagen. But all kind of this aggression works only if your skin cells are ready for this. And if you don't have fat tissue in your skin, if the fat tissue is resorbed with an age, or you just a little bit underweight, it can be difficult for your skin to react to the aggressive treatment. So all extremities are not so good for our body, for our health, but for the skin as well. So the best choice is to maintain the healthy weight, not be overweighted, but not be underweighted as well. That's how you can really prolong your skin active life and skin longevity. And all these 9 types of the skin, they are playing this beautiful symphony. And we should be very careful when we are addressing different skin cells type. For example, when you are using retinol-based prescriptions or retinol-based skincare, you are speaking the same biochemical language as your fibroblasts and as your keratoblasts. And you can speed up the epidermis renewal, but do you need it all the time? Because fast skin renewal means less of lipids on the skin surface, means less of proteins, including this useful opalescent, Elastin, or water-holding filgrane into the skin. It can activate fibroblasts, which can be collapsed in the same time. It will work, but sometimes it will work not as you planned to be, and it can create different kind of unpleasant or even dangerous side effects. Also, when you're using some pigmentation treatment, yes, it addresses the pigment cells, but in the same time, it can provoke the inflammation. And when the inflammation is provoked, it's provoked in the nearest neighborhood of the pigment cells. So when you irritate the skin, when you sensitize the skin, you activate the melanin production. So you can eliminate the melanin using the ingredient, but the same ingredient can reactivate melanin production and you can get even more of melanin in the skin. So your spots in the skin can become even darker in the treatment. So when you understand what types of skin cells you address, when you understand how ingredients and how different modality devices interact with different skin cells types, you can create really efficient and safe treatments. I'm really interested. Did you find this episode interesting? Did you find something new about skin cells types? If you have any questions, if you have any comments, please comment in the episode, comment on our socials, and I will be happy to answer and I will be happy to discuss all of it with you. Thank you for joining us today on the Meder Beauty podcast. If you found this helpful, be sure to subscribe so you never miss an expert skincare tip. Have a question or topic you'd like for me to cover? Send me a message at podcast@mederbeauty.com and let's keep the conversation going. Also, you can visit mederbeauty.com and discover Meder Skincare, which I created. And if you enjoyed the show, I'd love if you left a review. It helps others find the podcast and keeps us growing. Until next time, stay healthy, stay glowing, and take care of your skin.